Hello. Good afternoon, everyone. I'm just going to introduce today's speaker. And I have tons of things to say about him, so I just write it down just to be fast and let him present his data. So I'm going to introduce Professor Christopher Del Negro, who is a leading investigator in the field of central pattern generators, who got his PhD at UCLA, University of California at Los Angeles, with Scott Chandler working on intrinsic properties of mesencephalic trigeminal neurons, which are involved in different orofacial behaviors, including chewing. Once Christopher was in UCLA, two faculty professors there, Jack Feldman and Jeff Smith, were making a lot of progress in the identification and definition of the central pattern generator involved in respiratory rhythm generation. So he witnessed that development and decided to get a postdoc with both of them. So first he went to NIH to work with Jeff Smith, and then he moved back to UCLA to work with Jack Feldman. It's worth mentioning that when he was in UCLA, he shared the lab with Consuelo Morgado, who was a former member of our community. And after those postdoctoral stays, he went to the College of William and Mary to become a professor, where he has been making significant contributions to the understanding of synaptic, intrinsic, and population properties of the central pattern generator involved in the generation of respiratory rhythms. And I'm saying rhythms because you're going to see during his presentation that it's just not the normal breathing, but different kinds of rhythms. I also would like to mention that he spent a couple of sabbaticals, one in France with Gilles Sortan, learning about development and breathing generation, and another one in Denmark with Ole Kihn, who is a very important researcher in the studying the generation of locomotion. So then you can get an idea that he has a very good training and good knowledge about the physiology of CPGs. Finally, I just want to mention that his work has been constantly published in several prestigious journals, including Neurons, PNS, Annual Review Neuroscience, Annual Review Physiology, Nature Review Neuroscience, eLife, Journal of Neuroscience, Journal of Physiology, and so on. And he has already 5,800 citations with an age index of 36. So without further ado, I leave you with Professor Del Negro.
Thank you. Thank you for that kind introduction, Fernando. Thank you, Mauricio and Teresa, for inviting me to be here today. It's a great honor to share this afternoon with you and to celebrate the foundation of your Neurobiology Institute. I've been very impressed with you and your students in this wonderful building and this campus, so I'm really excited to share some of our research with you. So I'm going to pivot and begin to talk about the cellular building blocks of breathing. So neuroscientists, we are interested in how the brain generates behaviors, or when behaviors go wrong, they're pathologies. But what is behavior? Behavior is how an organism interacts with its environment. Very rarely do neuroscientists think about breathing as a behavior, but it's an important and an interesting behavior. So why is it important? Well, if you don't breathe, you don't live. That's the obvious part. But if something goes wrong with breathing at any stage of life, there's going to be a deadly or a morbid disease. So on the bottom here, I've indicated a lifespan from prenatal to a very aged person. And then these are a number of diseases which can affect an individual at any stage of life. All of them pertain to the neural control of breathing. In a conversation I had yesterday, we talked about obstructive sleep apnea and neurodegenerative diseases, and these can affect breathing and cause fatalities quite late in life. So breathing is important behavior. But breathing is also interesting behavior. Why is it interesting? Because we need to breathe in order to perform sports, dances, all of life's interesting elements. require control, precise breathing, and the physiology it underlies. But this is the intense use of breathing in sport, for example. But we also have a connection between breathing and our emotional lives. So this has been known for thousands of years in ancient Eastern traditions of yoga and meditation. And now recently, in some conversations I've had at the Institute here, we are more and more realizing that precise modulation and control of breathing can enhance brain function in general. So breathing is important. Breathing is interesting. And I'm going to make the case today that breathing may be the first behavior that we can understand at all levels of organization in the nervous system. Now, I know that's a bold claim, but let me try. Okay, so let's start with a very simple diagram of our breathing plant. This is the mechanical part of the breathing system, the airways and the lungs. And I want to start here because I'm going to build backwards from the breathing plant to the brain, how it controls it. So there are two sets of muscles. One is the airway muscles, control the airway and the patency of the airways leading to the lungs. And then there's the pump, largely the diaphragm, which is the bellows, which brings the air in and out. So let's go back and find the circuits that control breathing. So the first thing we'll do is identify the nerves, which is mechanosensation from the lung, and then the carotid sinus nerve, part of the glossopharyngeal nerve, which senses the blood gases and the pH and brings that information to the brain. Now, so far, this part is blank. I will reveal it now. That's the part that we study in my laboratory. This is the central pattern generator, which controls breathing. We are interested in the source of the rhythm, and then the circuits that turn that rhythm into an appropriate motor pattern to perform the behavior. So, for example, what the airways do is slightly different than what the pump does, and we need to have a pattern generator to control that. So I'm going to try and answer these 4 questions today. We'll start with the first, which is not my work, but then we'll move to 2, 3, and 4, which my lab has participated in. And indeed, most of this comes from the inspirational studies that Fernando and I did when we were postdocs. Mostly I followed that same route because, I don't know, lack of creativity, but I really stuck with this problem. Okay, one other thing that's really nice about breathing is it can be measured very easily using non-invasive techniques like plethysmography and piezoelectronics. Breathing can be measured very easily. This is the behavior of breathing measured in an adult mouse. Up and down means air goes in, air goes out. And when we study this today, we will understand the effect on the rhythm generator by changes in the frequency of breathing. So the rhythm generator is like a clock. The clock can go fast, the clock can go slow. We recognize those changes as frequency. And the pattern generator will be identified through changes in the amplitude. That's how we will study this behavior. So let's get started. Where is the site of rhythm generation? This is not my work, but the seminal work of Jack Feldman and Jeff Smith, both of whom I worked with as a postdoc. But we need to start with this so we know where to go next. Okay. So in the late 1980s and the early 1990s, our field discovered that one can remove the central nervous system from a newborn rodent, and it retains the ability to generate a breathing rhythm. So this is the neuraxis of a newborn rat. This is the rib cage. Nobody had cell phone cameras yet, so it's not a video. It's just a still picture. But if this were a video, those ribs would be moving up and down a fictive breath. But we don't have the video, but what we do have are nerve recordings from all of the efferent nerves that innervate breathing muscles. And they're rhythmically active. These are the ones that innervate the tongue and the airways, the phrenic, intercostals. These are all breathing nerves. And we retain this rhythm. So we can study breathing in the laboratory. Okay, now let's get to the site. If you reduce the size of the neuraxis, you can cut off the top of the pons and the medulla, and you can cut off the spinal cord and retain a very thin portion of the medulla. And in this region, we have this specialized region I've indicated in yellow called Prebotsum complex. This is the source of the inspiratory rhythm. This was a seminal work to come out of Jeff Smith and Jack Feldman's collaboration in the 1990s. The wonderful thing about this location is if we take a very thin slice of the medulla, not only does this medulla contain the Prebotsum complex, but it also contains pieces of the reticular formation and the hypoglossal nucleus, so we can continue to record the breathing rhythm from a very thin slice preparation. In other words, this slice contains a minimal model or a minimal microcircuit for breathing. You have the rhythm generator, and you have the pattern generator, and you have a nerve with motor output. So it's like breathing in a dish. And we can use this preparation, as Fernando and I have for many years, to study and interrogate how this system functions. Now just remember, the hypoglossal nerve normally innervates the tongue, and when we breathe, we just push the tongue out just a little bit each breath while the air comes in. So whenever you see the hypoglossal nerve discharge, it means inspiration, it means breathing in. Now let's move on to some of our work. We wanted to identify the cellular origin of inspiration. The Prebotsum complex is a region, but there's lots of cells there. It's a busy neighborhood. We wanted to find out which cells in this region are the source of the rhythm. And we used a genetic approach. So this picture indicates the developing neural tube, which becomes the spinal cord and the brain stem. In the developing state of an embryo, when the inner portion here represents the neural tube, there's a dorsal and a ventral orientation. The dorsal side will eventually become the sensory systems, and the ventral side will become the motor systems. And how the cells in this region develop their fate is determined by transcription factors. There's like a code of transcription factors which program this developing nervous system. I don't have time to show you all the reasons why we narrowed down into the transcription factor that we did, but I'll cut to the chase. We focused on DBX1. DBX1 is the transcription factor which gives rise to the canonical domain V0, or ventral zero domain. We focused on these for a number of reasons, and I'll begin to show you why. First of all, why DBX1 neurons? Well, it turns out they're rhythmically active in vitro. So I showed you a little while ago we can make this slice, and the slice retains the ability to generate a breathing-like rhythm. So first thing we did was we studied the neurons derived from DBX1 in these slices. So we'll use a tool, and I'm going to explain this tool very briefly. Of course, you've heard of Krelok's recombination, and we use Krelok's recombination a lot in my laboratory. So we have our workhorse. It's not really a horse, it's a mouse, but our workhorse animal is a DBX1 Cre mouse. So this animal will express Cre in every region where DBX1 is expressed. And then we cross this mouse with a lot of different reporters. The first one is just a TD tomato reporter. So there's a stop codon here, flanked by LOXP sites, and then TD tomato. So when we mate these two animals together, in the offspring, the Cre removes the stop codon and then expresses TD tomato. Now, of course, the whole mouse is not red, but just the cells which come from DBX1 precursors. So now I show you a picture of the slice, and now it's colored in red right here because this is the region where DBX1 cells form their population. The most high concentration is down here in the region of Prebotsum complex. So we'll make a recording. So we go with a high-power microscope, we find the red cell, we make a recording with a patch pipette, fill it with a green dye so we're sure it's the same cell, and we record it, and not only do we have the inspiratory rhythm, but we have these nice bursts. So we think these neurons, they're rhythmically active with inspiration, maybe they're important. So we study them further. Now, we have other reasons, but because we can't spend all afternoon together, I'll just make it really short. Number one, if you knock it out, they don't breathe. Okay? So that's supportive evidence. And then, number three, DBX1 neurons express the peptides that modulate respiration. So we are talking about neurokinin receptors, substance P, and we're talking about mu-opioid receptors and enkephalins. So we speed up breathing, slow it down. They're glutamatergic, and they are commissural, which means they project across the midline. Commissural is very important because we don't have left-right breathing. Breathing is just breathing. So the neurons which generate it, they must be equally connected on the left and the right, like the jaw. Not like limbs, but like the jaw, the diaphragm is bilateral. So we're interested in these neurons. So now how do we study and test their function? Well, we have a hypothesis. They make up the Prebotsum core. So we have two testable predictions. Number one, if you stimulate them, breathing should speed up. Second, if you inhibit them or silence them, then breathing should slow down or it should stop. Very simple. Very simple. Let's test them. Okay, we start with our workhorse again, but we're going to change the reporter. Now we're going to put in a channel rhodopsin reporter. It's called the catch. There are two stop codons, one with LOXP, which can be acted on by Cree, and then also a FRET-encased stop codon. So we put a virus in and make sure it's just in Prebotsum complex. And their offspring will express the channel rhodopsin and GFP. Okay, so now we did a lot of experiments in vitro, but I'm not going to show you those because we have time constraints. I'll just show you the in vivo experiments. So here's a mouse, and that's a ferrule, and I'm going to introduce light. Before I start the video, just watch the animal's abdomen moving, and then when the light comes on, observe what happens to breathing. So it speeds up. Okay, so that satisfies the prediction, but I want to make another point that you can't see from the video. I'm going to show you the traces now of that same animal breathing. As we increase the strength of the laser, not only does the breathing speed up, but the amplitude gets larger of each breath. That we did not expect. So it seems like stimulating the DBX1 core, yes, it affects the frequency, but it also affects the pattern. So hold on to that observation. Let's go to the next prediction. So the second one was about silencing or killing the neurons. Okay, so now we use our same workhorse. You should be bored of seeing this one by now. But we changed the reporter now, and now it's archaeorhodopsin. So this is the optogenetic construct which pumps protons out, so it's hyperpolarizing or photoinhibiting. And in the offspring, we have the archaeorhodopsin. So let's do the test. So I'm going to show you another video. This time the light will be orange light, and it will be acting on archaeorhodopsin. There's the animal breathing. Watch for the yellow light and watch its breathing. So that laser application was 30 seconds. A mouse will normally breathe 3 times per second. That mouse breathed 3 times in 30 seconds. So we were quite confident that we had the core of the Prebotsum complex, but again, we were very confused because not only did the breathing slow down, but the amplitude decreased a lot. So again, it seemed like it's having an effect on the rhythm generator, but also the pattern generator. Okay, now I want to show you an in vitro experiment to complement these. So here we go back to our slice. And now we use a multiphoton recording system, and we use it to map out the neurons in the Prebotsum complex, because remember, we can use a reporter mouse to make them red. So we use a microscope to find those cells, and then we record their locations in 3 dimensions. And then we create a matrix of their locations in the computer, and then we go back, measure the amplitude and the period of the rhythm, then we turn up the strength of the laser so that it can kill cells. Make the laser much stronger, 100% power. And then measure the amplitude and the period again after killing one cell and say, what happens? So here's the experiment. In this diagram on the top, I show you the recording of the hypoglossal nerve output. So that's the motor output of our system. And on the bottom, I'm measuring the period, or the cycle period of the rhythm. On the bottom is 0 to 20 seconds. And then it changes from 21 to 200 seconds. So this is all small cycle periods and large cycle periods. So when the experiment begins, we detect the targets and then begin to kill them. And the first thing that happens is the amplitude drops way down. And the second thing is that the cycle period increases. So again, by killing the DBX1 neurons, we have an effect on the frequency and on the amplitude, which suggests effects on the rhythm generator and the pattern generator. How do we make sense of this? We conclude that DBX1 neurons make the core of the Prebotsum complex, but they affect the rhythm and the pattern. Can we distinguish these two functions? So now we are going to try. So now I'm going to back up by some really good work by my colleague Jens Reckling at the University of Copenhagen. He did this work in France at Gif-sur-Yvette, but he's done it for many, many years. And he identified two electrophysiological phenotypes in the Prebotsum complex. One that he called type 1, he thinks they're rhythmogenic because they have pre-inspiratory activity. The neurons are early in their activity, seems like they could be leading the rhythm. And then the second one is called a type 2, and it has very little pre-inspiratory activity, suggesting it might be activated second, it might be part of the output system. So we take this idea and say, well, let's test to see if these are rhythmogenic and pattern related. So the framework for analysis would be this. We have the Prebotsum complex, and then we have the hypoglossal motor neurons here. So maybe we have rhythmogenic neurons communicating to premotor neurons, which communicate to the motor neurons. And then, of course, the axon goes out here. So we tested this notion in the laboratory, and I did this in collaboration with some good folks in the University of Alberta in Canada. So we make some recordings from type 2 neurons. Here's a patch clamp recording, and this is Prebotsum complex, of course, but this neuron is dorsal. It's on the dorsal extreme of the site. So we make this recording, looks like a type 2 neuron, and then we introduce an orthodromic stimulation. So you're making a patch clamp recording, you just give it some current. And then we introduce another electrode here in the nucleus for antidromic stimulation if the axons go there. Okay, so do something simple. Do an antidromic stimulation, stimulate the nucleus, generates an action potential, back propagates to the recorded neuron. Easy. Okay, now do two. The first stimulus is orthodromic. You give this with current injection in the soma. And then wait 25 milliseconds, stimulate the nucleus again. So now you have the first one propagates out, and then the second one propagates back. And they're separated in time. So now if we do them within 5 milliseconds, now the orthodromic starts moving out. You stimulate the nucleus, this one comes in, and they collide. And so it proves that they're connected. So we showed in this study that we could have DBX1-derived type 2 neurons which project out to the hypoglossal nucleus, forming a pattern-generating circuit, or premotor neurons. Okay, so let's test this further. We went back to our slice preparation and our multiphoton recording system, except now we move the target. The target is not the Prebotsum complex, but dorsal, on the dorsal side where we expect the premotor neurons to be. We use the same approach, except we also keep the target only on one side. So now, when we laser ablate the neurons, on the right side, the amplitude goes down, but on the left side, there's no effect. And there's no change in the cycle period. So in this experiment, we show that there is a rhythm generator and there is a pattern generator. And the rhythmogenic neurons are the type 1, and the premotor neurons are the type 2, and they're dorsal. So now I want to pivot, and I will begin to talk about the mechanism. This is a very difficult problem. When Fernando and I got into this field, it was arguments all the time about the mechanism. And I think it's taken 25 years to get to some understanding. We all agree excitatory neurons are the most important part, and synaptic interactions are the most important part. But what I'm showing you now is still a hypothesis of how the rhythm is generated, and we call it the burstlet hypothesis. This hypothesis was started by my good friend and colleague Kai-Wen Kam. He's now in Chicago. And my group subsequently picked up Kai-Wen's ideas, and we tried to do a little bit more with those ideas. So let's talk about the burstlet hypothesis. So the idea is this. In the Prebotsum complex, the excitatory DBX1-derived neurons, they communicate with one another, and they make a little network oscillator. So they oscillate among themselves as a network. And we call this a burstlet, because if nothing happens, it's only happening in the Prebotsum complex. However, occasionally, if the burstlet reaches a threshold, If it reaches a threshold, then it can trigger an output from the Prebotsum complex, and that's a burst. So the idea is that there is a burstlet mechanism, and if it reaches threshold, it becomes a burst and an output. So the rhythmogenesis occurs in the burstlet, and the output occurs with the burst. Bursts are essential for breathing because if the information never gets out, then the animal never takes a breath. However, the idea in burstlets is that the burstlet itself is rhythmogenic. So this idea, through today's talk, the idea is that in the Prebotsum complex you have rhythmogenesis and you have pattern output. And they are functioning at the same time in the same place, although it appears as though two different cell types are responsible. So let's test this idea. In our in vitro case, when we have this rhythmic slice, we keep the excitability high. In the laboratory, we do this by putting 9 millimolar potassium in the bathing solution. So for those of you that work with slices in the laboratory, you want to mimic the brain's milieu. That may be like a potassium level, more like the level of 3. But in the respiration work, we often take the potassium and raise it up to 9 because all the rest of the brain is gone, so we have to keep excitability high by just manipulating potassium. So when I write high excitability, I mean this slice is being bathed in 9 millimolar potassium, so it's high excitability. Under these conditions, you have lots of bursts, and you have lots of bursts in the Prebotsum complex field recording. So this is a normal situation. However, occasionally, we recognize this thing like a burstlet. In other words, there was some local activity in Prebotsum complex, but it never got out. It never created a burst. Now, if you lower the excitability, now we will do this by lowering the potassium down to like 7 or 6 millimolar. Now, you still have bursts occasionally, but now you see more burstlets. Because as you lower the excitability, the ability for the system to generate the output is diminished. And I'll show you the whole spectrum from highest potassium to lowest potassium. As the excitability goes down, the fraction of burstlets goes up. The system remains rhythmically active, but it generates fewer bursts as the excitability goes down. So this suggested to us that there's a rhythm generation, and then there's a pattern output. So could this really be the source of the rhythm? Well, one necessary test is that it must be happening on both sides. Because if I show you a recording and show you a little burst that you say, well, I don't know, maybe it's just some random activity, I don't know, it doesn't look meaningful to me. Well, if it's meaningful, it has to happen on both sides at the same time. Remember, breathing is synchronous on the left and on the right. So we look for that. We make Prebotsum recordings on the left and the right with the hypoglossal output, and you have bursts. They're, of course, bilateral, but the burstlets are almost always bilateral. That's the only exception. All the other ones are happening at both sides at the same time, so it suggests it's a viable mechanism. So what gives us the sense that this is rhythmogenic? Well, let's just do a cycle-triggered average. So we recorded lots of burstlets, and then we averaged them together, and they have this nice shape. Then, when we aligned this average with a cycle-triggered average of the bursts, we noticed their slopes look exactly the same. So it suggests like the upswing, the initial part of the burstlet, and the initial part of the burst is the same thing. And when there is motor output, the motor output doesn't happen until the burst picks up from the burstlet. So it suggests that the burst is what brings this activity and moves it out to the motor neurons. So we think the rhythm is here in the pre-inspiratory phase, and the inspiratory and the burst are motor output. We think the mechanism is the synaptic interactions among the neurons in Prebotsum complex, because if you look at any one burstlet, you see a summation of EPSPs. So it looks like it's the synaptic interactions. And we are studying this, and other groups are studying this with models and more recordings. We don't have the luxury of time to go into those models now, because I want to hit on a number of different topics. But let's do one more thing before we go. Let's look at this dichotomy, separation of rhythm and pattern. So now we go back to our multiphoton system, and we use a DBX1 G-camp mouse. So I don't show you the whole diagram again. You've seen it like 7 times. It's enough. This time we have the same DBX mouse, but we are crossing it with a G-camp reporter system. So here I show you motor output, and here's 5 individual neurons recorded with multiphoton. The blue squares show you the burstlets that are occurring in those neurons. Notice it's neuron 2, 3, and 5 that have burstlets and bursts. But neurons 1 and 4 are just active during the bursts. So we think these ones are the rhythm generation, and these ones here are just the motor output. So okay, now let's change excitability. We go to low excitability. Look, it's still 2, 3, and 5 that are generating burstlets. And now they have more burstlets because the excitability is low. And 1 and 4 are still generating the bursts. So from these experiments, we conclude that the burstlet hypothesis is not proved, but it's a viable hypothesis to explain inspiratory rhythm generation. And we conclude that there are 2 populations. The rhythmogenic ones are DBX1-derived V0V or type 1 neurons. And the ones that generate the output are also derived from DBX1, but they are V0 dorsal or the type 2 neurons. And this is our framework for understanding rhythm. So I want to pivot now, and I want to go and talk a little bit about the ion channels that generate the output. And here I want to mention my colleague Fernando again, because we met in the year, I think, 2001, and he was beginning to study a very important current that I've spent like 15 years working on now, the CAN current. So we're going to talk about the CAN current a little bit. So we have this idea that the rhythm generation is due to the burstlet, the pre-inspiratory phase. Well, while the neurons are engaging in this activity, calcium is increasing in the cytosol because, well, the neurons are spiking and the calcium channels are opening and calcium's coming in. It's obvious. But what happens with that calcium is it seems to activate a calcium-activated sodium-mediated current that we call CAN current. And that seems to create this very strong burst, this CAN current. And this is work done in lots of groups. We became interested. What turns the burst off is probably synaptic depression and sodium pumps. I will not have the luxury of time to talk about this today. But I really want to focus on the CAN current. So when we began to study the CAN current a long time ago, it's just a phenomenological current. We talk about it at the whole cell, but there's no such thing as a CAN ion channel. We have a CAN current, but there's no such thing as a CAN ion channel, and nobody really knew where we would find the ion channel. And this is what took us a long time to figure out what is the ion channel to test the role of CAN current. So we started to use a different approach, and this is one which involves genetics. So we did RNA-seq, or RNA sequencing. So to do this, we make a slice preparation, and this is our same workhorse mouse with a TBD tomato reporter. We go into the Prebotsum complex, find the DBX1-derived neurons, and then pick them out. We call them POS for positive for DBX1, and then neg for any old negative cell which is not derived from DBX1. And then we're going to compare their genomes. So we take these, lyse them, create RNA, make a cDNA library, analyze it for quality, send it out for sequencing, and then study the transcriptome of the DBX1 versus the non-DBX1 neurons. Now, of course, we had 27,000 genes, and we don't have enough time to talk about 26,993 of them, but I'll talk about a couple. So we'll talk about the ones that come from the TRP channel family, transient receptor potential. Our idea was that these TRP ion channels activate due to a lot of different stimuli, one of which is calcium. So we focused on these two, TRPC3 and TRPM4, which can be activated by internal calcium. Although TRPC3 has a higher expression level, it turns out that TRPM4 is the one that's more important. And so I'll skip a few details and go right to those tests. Here's the best immunohistochemistry I ever did in my life. And we found that in the DBX1-derived neurons in Prebotsum complex, they express TRPM4, and there's the merge. So not only did we find this TRPM4 gene in the mRNA library, in the transcriptome, but we also found evidence for the protein in our DBX1 neurons. So now we're going to test the role of this ion channel. Here's how we do it. We use a short hairpin RNA. So a short hairpin RNA is an intervention which can suppress or diminish the expression of RNA in a cell. So we designed an AAV expression vector which has a payload of shRNA. It gets into the nucleus, releases its DNA. It creates a short hairpin RNA because it looks like a hairpin. This eventually will become a short inhibitory RNA, and it will degrade the TRPM4 RNA and make it non-functional. That slide went by a little fast. But the shRNA goes in, and then it's able to degrade the function of TRPM4 RNA. So now I'm going to take you through an in vivo experiment. And as I told you a few minutes ago, all of these experiments have in vitro components, but because of time limitations, mainly I show you the in vivo experiments because they're a little bit more convincing because it's really breathing. So I'm going to show you the baseline breathing and then show you breathing in subsequent days after the shRNA is injected. So here's the beginning, nice breathing pattern. Day 9, I don't know, maybe it's different, maybe not, I'm not sure. Day 15, I start to see that the breaths look smaller. This VT means tidal volume. The volume of the breath that comes in and goes out. Tidal means like you have the ocean, the tide goes in, the tide goes out. So we think of breathing the same way. The tidal volume is the volume of breath in, breath out. Well, the breathing is getting smaller. But what you may also notice is that the breaths are more frequent. So again, we have rhythm and pattern. What's happening here? This is a living mouse, not anesthetized. It's living for many days. So the tidal volume is getting smaller, and the animal still needs air, so it's breathing faster. It's breathing faster to make sure he has enough air. But after a month, he really can't keep up. And there are gasps and what we call ataxia, which means the breathing is not good. The pattern is no good. And by the end of the study, the animals die of respiratory failure. Now, if we look at the progression in these plots here, days after shRNA, the tidal volume goes down. And the frequency goes up because the animal's trying to breathe faster to maintain its physiology. But the density of the dots goes down because the animals are dying as the study goes on of respiratory failure. So from these studies, we conclude that TRPM4 ion channels form the CAN current, and they mediate bursts and output. So they are essential for generating the output. The reason the frequency increases is because the animal is consciously doing it. But we think that really the importance of this current is to generate the output. So the next thing that we want to do is say, can we learn anything else about these neurons from their transcriptomes? Because we think we understand we have the type 1 generating the rhythm, the type 2 generating the output. We think we understand what the CAN current is useful for. But what about other differences between these neurons? So to try and get at that question, we go back to a genetic approach. We're going to slightly change things. Now we're going to do what they call patch-seq. So now we're going to patch clamp the neuron before we sequence the transcriptome. It's a little bit more complicated. In fact, it's a lot more complicated. It took my very, very talented graduate student, Prajita Kalukur, it took her 1.5 years just to generate the samples. So we make a patch clamp recording from our slice, and then we look at this recording and we say, is it a type 1 or is it a type 2? And after we know what type it is by the recording, then we extract the cell, aspirate the contents, make a cDNA library, make sure it has high enough quality, send the library out for sequencing, and then do the bioinformatics when we get the sequences back. Now, again, these studies, they generate thousands and thousands and thousands of genes, and we have a limited time together. So I want to highlight just a couple little things that you may find interesting we learned from this study. Here is the list of the top 123 differentially expressed genes. At the top, in magenta, are ones that are expressed more highly in the type 1. And then in the blue, these are the genes that are more highly expressed in the type 2. And I'm just going to highlight 2 things. 4 genes. This one, this one, this one, and this one. Now, they're totally small, so I'm going to blow it up for you. Here's GRIA and GABRA1. So GRIA is AMPA receptors. And GABRA1 is GABA receptors. And these are more highly expressed in type 2. From this, we interpret that the output neurons, they express more highly the synaptic receptors because their job is to receive the signal and push it on. So they have a higher expression of synaptic receptors. That's our interpretation. The next one, also in type 2, SGK1 and cracker2A. These have to do with the scaffolding that sets up calcium entry and to the CAN current. So bringing calcium into the cell, storing it, releasing it, and activating the CAN current. Again, these are more highly expressed in the type 2 because, again, we think that this is more important for generating the output of our system. So the synaptic receptors and the CAN current, very important for output. The last thing that I'm going to highlight is something that is very important for society. Right now, in the United States and in many parts of the world, the opioid epidemic is killing recreational drug users by the thousands. And these drugs kill people because they make them stop breathing. So we want to understand a little bit more, and many other groups want to understand, how do opioids affect the breathing center? Well, what we noticed here is that the magenta plot and the blue plot, this is type 1 and type 2. They all express preproenkephalin. So they express the enkephalins, which is the endogenous ligand for opioids. However, the pink bar for OPRM1, opioid receptor, mu opioid receptor, is much more highly expressed in the type 1 neurons. They are the source of the rhythm. So if opioids are affecting in the Prebotsum complex, they're hitting the rhythm, and that's why the breathing slows down and stops. Of course, it's not the whole explanation. It's just a tiny part of it, but that's one of the things that we learned from this study. So it's not that the output is the problem, but the rhythm is the problem from the opioids. So we conclude that the synaptic receptors and the CAN current are important in the output neurons, and that mu opioid receptors, although they are sparse in the Prebotsum, are expressed in the type 1 neurons, and they affect the rhythm. So I want to do some interim conclusions before I talk about the neural generation of Sigh. So the Prebotsum is the inspiratory oscillator. Not my work, but it's an important part of today's talk. This is Feldman and Smith. But this is our work. DBX1 neurons form the core of the Prebotsum complex. They are inspiratory rhythmogenic, and they're also important for the output pattern. We think the rhythm emerges due to a burstlet mechanism generated by type 1 neurons that synapse on type 2 neurons to generate the output. And that output is important because the TRPM4 ion channels making up the CAN current help produce this strong output. So these are the main conclusions from the first part of the talk, and I promise the second part will be shorter, because it's going to be a very short story. Okay, let me get a quick drink, and then we move on. So breathing consists of the normal eupnea. We breathe in, we breathe out for physiology. But we are mammals. Mammals are unique because number one, we have the diaphragm. We are unique because we have a diaphragm. But secondly, our lungs have collapsible air sacs called alveoli. And those alveoli dramatically expand the surface area for gas exchange in the lung. And that's wonderful to have so much surface area for gas exchange, because it means we can have much higher metabolisms to support our oxygen-hungry brains. But there's an Achilles heel. There's a problem, because the alveoli can collapse. And if they collapse, then that surface area goes to waste. So mammals, have the unique breathing feature called a sigh. And a sigh is a very, very large breath indicated by a Greek sigma. These are just breathing traces of 10 different mice. The little small wave is just normal eupnea breaths, and then every once in a while you see a big breath with sigma. That's the sigh. Whether you realize it or not, every 3, 4, 5 minutes, you take a very large breath, which makes the alveoli expand. Our surfactant redistributes, and then our breathing surface area is maintained at its large state. And if we don't sigh, our lungs will slowly collapse and get smaller and smaller. And that can be very pathological. So we need to sigh. Sighing is important. Now, even if we go back to the year 2000, Nino Ramirez's lab recognized that the Sigh comes from the same place as Eupnea. It comes from the Prebotsum complex. But it didn't tell us how it's generated or what the mechanism or which cells do it. So that's where my lab started to work on the problem. We wanted to understand what gives rise to the Sigh. What are the cells and what is the mechanism? So the first thing we ask is, are there really separate mechanisms? We'll go back to the slice. Under high excitability conditions, 9 millimolar potassium, we have these normal Eupnea-like bursts, and then every once in a while we have the Sigh, which is indicated by sigma. It may be difficult to see on the video screen, but these Sigh bursts, they look thicker, right? They look a little bit thicker. They're larger. There's more the area under the curve is bigger. They're a bigger burst. When we lower the excitability, the Eupnea rhythm, it slows down and eventually will stop, but the Sigh continues. So now let's look at the relative change in frequency as we change excitability. On the x-axis here is the potassium level, so high excitability and low. The inspiratory frequency decreases as excitability goes down, and the Sigh frequency goes down too. But by looking at this plot here, we plot Sigh frequency on the x-axis, inspiratory frequency on the y-axis. This curve has a slope of 20, which means as we change excitability, you have 20 times the impact on the inspiratory frequency compared to Sigh. So the inspiratory frequency is much more sensitive to changes in excitability. Okay, let's ask an inverse. Can we exclusively modulate Sigh? So now we do the same thing in a slice preparation. These are the Sighs, and now we add neuromedin B. Neuromedin B is a bombesin-like peptide which affects the Sigh system. And this is the work from Kai-Wen Kam and Jack Feldman. When we administer neuromedin B at 10 or 30 nanomolar, the Sigh frequency increases, but the inspiratory frequency does not change. So there's NMB concentration. Inspiratory frequency is flat, no effect. Sigh frequency increases. And now if we plot it the same way, as we change the concentration of NMB, Sigh is affected a lot, its frequency speeds up, but there's no effect on inspiratory frequency, slope is 0. So this suggests that there are distinct rhythmogenic mechanisms at work. Well, people have been thinking about this for a while, and some of the leading ideas were that what if there are two populations of neurons in the Prebotsum complex generating these two different rhythms? They can be separate. Okay. The other one was that, well, the Sigh frequency is so slow, maybe it's generated by astrocytes. Maybe it's due to glia. So we tested these ideas by going back to the multiphoton system, where we can record lots of neurons at the same time. These are all DBX1-derived neurons. And what we see is that the neurons are all rhythmically active, and they are all rhythmically active in the presence of the Sigh as well. So it suggests that both rhythms are active in the DBX1 neurons. And then, when we use a cocktail of astrocyte antagonists, which prevent communication between astrocytes and neurons, there was no effect. No effect on inspiratory frequency and no effect on Sigh frequency. So from these experiments and some other complementary experiments, we conclude that the Sigh rhythm and the inspiratory rhythm, they probably come from the same population of DBX1 neurons. Now this is unpublished data, still in bioRxiv. So I care about you enough to show you the unpublished data. And I think it's on YouTube too, so I guess I'm telling everybody. All right, so we wanted to see, is it possible for one population to generate two rhythms that are so separate? So for this, we made a mathematical model. And this I did in collaboration with a good friend of mine at William and Mary, Greg Conradi-Smith. Looks like really complicated stuff, but in fact, you will understand it very, very clearly. The first three equations are just generating the inspiratory Eupnea rhythm. This A means neural activity, spiking rate. And S means synaptic depression. And theta means activity-dependent outward currents, like sodium pumps and so on. This will generate a Eupnea-like rhythm. Then these two equations give rise to the Sigh rhythm. Now these look really complicated, but I'll explain them in the next slide. That's cytosolic calcium, total calcium. This is a picture of a neuron signaling mechanisms. And I left out Hodgkin-Huxley currents because all neuroscientists understand them already. You don't need to see a picture of it. So the first term here, J out, represents the plasma membrane calcium ATPase pumps. That's just a Hill function, pumping calcium out. Second term, J in. This just means the leakage through calcium channels and the voltage-dependent activation of calcium channels during spikes. That brings calcium in. Third term, J circa. It's same thing. It's a Hill function, and it represents the circa pump, sarcoplasmic endoplasmic reticulum out ATPase pump, the circa pump. And then the last one is the release rate through IP3 receptors. So each of these terms is well established biophysically. So now we put this system into motion and we generate two rhythms at the same time. So that's the Eupnea rhythm, and then the big one is the Sigh rhythm. Now, over here we show theta and S. Imagine we begin the interburst interval here when theta is high. That means you have a lot of outward current. The pumps are full blast. You have a lot of activity-dependent outward current. And then gradually, as the interval goes on, this curve drops down because those outward currents deactivate, and S gets larger because the synaptic depression recovers. When we get here, there's an inspiratory burst, and then during the magenta phase, synapses depress, they go back down, and the outward currents activate, so we head back up. That's one inspiratory cycle. Now, as this is going on, as these cycles are going on, the calcium is increasing very, very, very slowly. Because each time we go through an inspiratory cycle, a little bit of that calcium gets sucked up into the endoplasmic reticulum. And that's at the bottom curve here. That's total calcium. The total calcium is increasing little by little by little by little by little. And then when it reaches this point, it releases everything. It releases all that calcium. And that activates the CAN current and then gives us that really big burst. That's the Sigh. So that's just a mathematical model. We wanted to test this. The mathematical model says one population can do both rhythms. First thing we did was test those excitabilities. On the left is the experiment. These are the two graphs I showed you already. Inspiratory frequency versus Sigh frequency. As we change excitability, you have much more effect on inspiration, slope of 19. Manipulate with NMB and the slope is flat. It only affects Sigh frequency. Now we do those same experiments in the model. So manipulate the excitability through this parameter, and we have effect on inspiratory and Sigh frequency, but the slope is 20. It's far more impactful on the inspiratory frequency. And then we manipulate the IP3 receptors, which is what NMB does. And under these conditions, the slope is 0. No effect on inspiration, but the Sigh frequency is accelerated. So these suggest that the model can also generate these separate rhythms, and they can be regulated separately, even though they're coming from the same population. Why? Because you have two tool kits. One generates inspiration, the other one uses intracellular signaling to generate Sigh. Now, is the model correct? Well, let's make some more predictions. The first one is this, and every modeler says they do the predictions first and then they do the test, but it's usually not the case. They usually do the test first and then manipulate the model so its predictions are right. But I swear to you, we did the predictions first. I promise. Here's control. Now, in the model, we block. This is a model, right? So it's not really a circle block, but it's a model of a circle block. If we block it by 50%, the Sigh rhythm speeds up. And if we block it by 80%, well, it's too much and the system stops. So here is a recording. There's the Sigh rhythm. When we give a low dose of thapsigargan, the Sigh speeds up. So that looks like the first case, where the Sigh speeds up. And then, when we increase the dose tenfold, then it stops and doesn't come back until washout. So that suggests like the second one. So our first prediction is, if you stop the endoplasmic reticulum from taking up calcium, you should speed things up and then stop it. The other prediction has to do with the IP3 receptors. So here, if you block IP3 receptors by 40%, it should slow the Sigh rhythm down. If you block them 50% or more, it stops it. This is a really tight prediction. It's hard to get a drug to mimic a 10% change. So we use this really good drug called zestospongin, which is produced by coral reefs. It blocks IP3 receptors very selectively, But we couldn't really mimic a 40% or 50% block, so we just gave this drug and it always stopped the rhythm, suggesting it goes right to this point. So it stopped the rhythm. So our conclusions from these studies, one population of neurons can generate two rhythms. One for eupnea or inspiration, and the other one for the sigh. These rhythms depend on separate tool kits. One involves voltage-dependent network properties to generate inspiration, and the other one depends on intracellular signaling and calcium. And we think that as breathing evolved, breathing started with fish, and then as animals moved onto the land, they developed compliant lungs, but still breathing was inspiration and expiration, inspiration and expiration. But when we get to the mammal, we have the collapsible air sacs, and we have the diaphragm. And now, suddenly, the breathing plant needs a new rhythm, because inspiration is not enough. It needs something to reinflate those alveoli. But the brain has been evolving all the way through fish and amphibians and so on, needs a new rhythm. Where does it get it? Well, the brain has lots of options, but in our opinion, our hypothesis is the brain said, well, maybe we don't build a new rhythm generator. Maybe we just adapt the pieces that we already have, like a calcium signaling pathway is already there. Let's just modify it slightly, and then by doing so, it can generate a second rhythm from the same population of neurons that's already generating the inspiratory rhythm. And our model shows that it can work. And not only can it work, but you can still regulate both rhythms separately for different physiological purposes. Now, I started the talk by claiming that breathing may be the first behavior we can explain at multiple levels of organization in the nervous system. And I think that's true, and I hope, even if you're not fully convinced, you'll think, we're pretty far along in understanding breathing in this pretty exciting system. But I want to spend 1 second, as I finish, to say, what's next? What's next? If we do figure out how breathing is generated and regulated, we figure all this out, what do we do next? Do we just stop and retire? No. I think the inspiration comes from what some of you are already doing, including in Fernando's lab, to say, what else is the brain doing with the breathing rhythm? Well, people are beginning to look at how the breathing rhythm can be, in a sense, utilized or hijacked to perform other computational functions in the brain. This is just beginning. I think this is the most exciting wave of work that will emerge in the next decades as we finally solve this problem. And with that, I want to thank all these great people that were involved in these studies, and I want to thank you again for your attention. And if anybody is interested in graduate or postdoctoral studies with me, please contact me by email. I would love to talk to you. Thanks for your attention.
Hi. Thank you very much for the wonderful talk. I have two questions. One, I think both are related, but the first one is, I saw in your optogenetic experiments in the DBX mice that when you decrease the breathing, I was looking at the, let's say, rebound breathing, and I didn't see an increase in frequency or amplitude after you decrease the breathing by using archaeorhodopsin. So my question is, what's the relationship of the oxygen levels to this particular system? Because I was imagining like, the animal goes without breathing. I thought he's going to die. And then I thought he would start breathing faster or with more amplitude.
In those particular traces that I showed you, the breathing was only arrested for 5 seconds. So I think it was not enough time to see it in the traces. However, if I were to show you the actual trace from the 30 seconds, you will see a very strong rebound. That's a very shrewd question. I think in the traces that I showed you, it was just too short to make the metabolism change enough to cause that rebound that you said. But if we have time, and we'll be in email contact, I know, I'll show you some of those traces.
Very good. And then the second is related because this Prebotsum is close to the locomotor region, of mesencephalic locomotor region. And
no, it's not very close. It's below facial nucleus.
I see.
Yeah, so it's quite a few millimeters away from it. But it's
Okay.
near the reticular activating system, like gigantocellularis.
I see. So, but in any case, when you run fast or slow, I guess your levels of oxygen go down. And I was thinking, what's the relationship between these two systems? If it's on the direction from the locomotion that will make the animal or this Prebotsum go faster, or would be the other way around?
Your instinct is exactly right. And right now, this exact question is being studied by my colleague Julien Bouvier in France. But in fact, during exercise, oxygen levels never drop. What happens is the carbon dioxide levels increase. So there seems to be two things that stimulate faster breathing. A little bit higher carbon dioxide, and then also the sensory feedback from the movement of the limbs comes up through the spinal cord and also tells the Prebotsum to breathe faster.
I see.
Yeah.
Thank you.
Yeah. But this work is, like I said, this is ongoing with Julien Bouvier.
Hi, I wanted to ask about the origin of these cells. You were mentioning that they were DBX1 positive, but clearly there were many other cells around. Have you delved more into that to see whether there were other genes that are expressed that cause them to differentiate into these two different populations in the Prebotsum complex, or?
I think if I understand your question carefully, you said, so DBX1 is a good marker, an indicator for this population of respiratory neurons. And are there other genes that are involved in differentiating the neurons into rhythm and pattern generators? We are still working on that problem. Because we did our patch-seq studies and our RNA-seq studies on mature neurons, all these developmental transcription factors, they're all gone. So now we're beginning to do embryonic level RNA sequencing, and we want to find the more genes that differentiate them. But we don't know the answer yet.
Thank you.
Hi, Christopher. Thank you very much for your beautiful talk. I know that olfaction can affect breathing, and I don't know if olfaction can affect these neurons, the ones that are having the rhythmicity. But thinking in what you said at the end of your talk, so it's possible that the rhythmicity or the activity of these Prebotsum neurons affect also olfaction or affect the brain in some other ways. Can you elaborate a little bit about this idea?
I do not know whether olfaction can affect Prebotsum complex, and I don't know if anyone has worked on it. However, Christine Zelano and several other of her colleagues are looking at how air passing over the olfactory epithelium is affecting the ability to make memories, consolidate memories, and recall memories. There's another paper I think I discussed with a little bit earlier today about Sirota and some colleagues looking at how air passing over the olfactory bulb can affect brain processing in general. It's very exciting new work, but I am reluctant to speculate too much on it because it's other people's work. But I know for sure it's happening.
I'm going to make some questions. Regarding these burstlet cells, I'm going to call them burstlet cells because you find some that are pretty prone to participate in burstlet. Are they more connected with each other in contrast with other types of cells in the network?
Fernando, I wish we knew the answer to it. We did 1.5 years of double patch recording, and we very rarely ever found any neurons connected to one another. So it's a losing game to try and record two neurons at the same time and find connections. Boy, we just did not succeed. So I don't know the answer, although I wish I did.
Okay, and then you found that these type 2 cells, which are kind of responsible for the motor output, have more receptors both for glutamate and GABA. Have you just made the corroboration of applying either glutamate or GABA and getting bigger responses in these cells compared to the type 1s?
We have not done it. And we finished the patch-seq study, and then we moved on to different questions, but we could do that, but we have not done that to test whether the higher expression of the genes results in a higher expression of the protein in terms of the physiology, although it's an obvious next step, but we have not done that.
Okay.
But I think probably there may be differences in input resistance to consider too.
Okay. And I just want you to elaborate on some observations that and several laboratories have made throughout the time, that every single time you apply something that depresses the activity of the Prebotsinger, not just on metabolic manipulations, but also on intrinsic properties. Every single time you see a depression of Sigh right away, before any alteration in Eupnea. So that makes some problems for me in order to understand these two mechanisms that you are proposing are kind of working in parallel.
My initial guess would be that the inspiratory rhythm is essential to help get the calcium inside. So if you have some perturbation that will affect excitability in the Prebotsinger complex, then its initial effect may be to slow down the uptake of the calcium. But I'm speculating because we didn't test it thoroughly. But our guess is with the model, we could probably try that prediction, affect the parameters of excitability, and see if the effect on the Sigh is initial. So it's testable.
Cool. Thank you.
You're welcome. Thank you.
Another question?
This is not a mechanistic question, but more applied, let's say. So I've heard that hypertension can be now treated with a machine that teaches you how to breathe deeply, a respirometer or something like that, and it's been approved by the FDA. So I'm wondering whether these Prebotsinger neurons, because of our way of life, sedentary lifestyle, and the way we have changed our feeding practices, has somehow affected this pattern of breathing, our breathing behavior, that now we need to learn again how to breathe properly, and now we are affecting our heart and other systems. So I want to hear your comments on this.
I agree with you 100%. I think your hypothesis is correct. And I think when I first practiced yoga a few months ago, I started maybe 7 months ago practicing yoga, and I didn't want to tell my teacher that I study breathing because she would say to me, just now you start yoga? I mean, we've known this for 3,000 years and you've been studying breathing for 2 decades and now? I felt stupid. But I think you're correct. Now, I don't think that the origin of the health problems you're talking about is coming from Prebotsinger. I think it's all the systems that are regulating Prebotsinger. So, for example, the most simplest breathing discipline to help relax the body and relax the mind is take an inhale that's maybe 2 seconds, and then exhale for more time, 4, 6, 8 seconds. Why? Because when you breathe in, you stimulate the sympathetic nervous system, exciting the brain. And when you breathe out, you stimulate the parasympathetic nervous system via the vagus nerve, and it tends to calm you down. So if you take a few minutes and make short inhale, long exhale, after a few minutes, I guarantee you, your physiology is based on it, you will feel relaxed. And I think that, at least from a neuroscience perspective, that's the way it's working. But I agree with your hypothesis.
Okay, thank you.
Any other question? If not, thank you, Christopher, very much.